Hello, and welcome to part 4 of the Innate Immune chapter. In this section, we're going to talk a little bit about cell communication. In particular, we're going to talk about the different chemicals that the immune system uses for immunological communication between immune cells. The immune system does need to have its own specific language, and in order to understand that language, we do need to understand a little bit about how cells in general communicate. Now, cells communicate through chemical signals, and so on the surface of cells, we have a receptor protein. And this receptor is think of it as serving as an antenna. Now, a cell over here, this is a process known as endocrine signaling, which most immune systems, most signaling systems in the immune system will use. And that is when we signal from one cell far away to another cell. And this is done through chemicals. So these cells will excrete molecules, chemical molecules. These could be proteins, these could be compounds, these could be acids or bases or any kind of chemical compound or molecule that's released by the cell. This molecule will enter into the immune system and eventually make its way to another cell. Now, any cell that has this receptor for this molecule here will receive the molecule. The molecule binds to the receptor. This then sends a signal to the inside of the cell, which activates or does something to some kind of transcription factor to initiate a cellular response. That cell will respond by perhaps sending out a signal of its own, changing shape, carrying out an action, or some type of effect. Now, the molecule, the signaling molecule, is known as a ligand or ligand. And of course, this purple thing here that is receiving the signal, the antenna, is known as the receptor. So this process is known as a receptor ligand complex. So these cells, all cells, have to receive a signal to their receptor, usually to carry out some sort of activity. Now, different classes of signaling. Endocrine signaling is when we are signaling from far away. Autocrine signaling is when a cell is releasing a compound that in turn signals itself, so it's giving itself a signal. Paracrine signaling is when it is signaling a cell that is close by, so from one neighbor to another. And then contact-dependent signaling is when two cells must come into actual contact to talk to each other. We are going to see literally all 4 of these different mechanisms of signaling in the innate and adaptive immune system. Endocrine signaling we will see with chemotaxis. In receptor ligand autocrine signaling here, in autocrine signaling we will see this in the fever response. In paracrine signaling, this is going to occur between B cells and T cells. And in contact-dependent signaling, this will occur between antigen-presenting cells and T cells, also between T cells and B cells and other cells involved in the immune system. So what are the different molecules that the immune system uses for talking? They actually have, the immune system has its own chemical language that some other cells will recognize and some will not. So chemokines are responsible for chemotaxis. This is the attraction of white blood cells or other immune cells to a site of injury or infection. So these cells have to send out a chemokine signal is used to initiate chemotaxis, to recruit neutrophils or T cells or antibodies even. Another class of cytokine is the colony-stimulating factors. Colony-stimulating factors are normally produced by cells that have been damaged or are trying to repair themselves. So colony-stimulating factor initiates proliferation, meaning cell growth, and differentiation. This particular cytokine here is used in healing. Think of it that way, right? In healing. So to promote new growth of skin, new skin growth, and new tissue growth to repair and replace that which was damaged from either infection or injury. Next are the class of interferons. Now, interferons, there are 3 basic families or classes of interferons. Interferon class 1, class 2, and class 3. Both class 1 and class 2 interferons are involved, have antiviral properties. So these are involved in virus responses. Class 3 are interferons are produced when intracellular bacterial infections occur. And for the sake of this class, we're going to be talking mainly about class 1 and class 2. Also, the interferons can be regulatory. Interferons can stimulate other cellular activities and also inhibit some cellular activities. It all is going to depend on which cell those interferons interact with. Interleukins are probably one of the largest classes of cytokines in the immune system. There are 17 known families of cytokines. Interleukins. Just interleukin 1 has about, I believe, 7 or 8 different subclasses of interleukins, IL-1A or IL-1 alpha, IL-1 beta. So there's lots and lots, over 50 different known interleukins. Interleukins are produced by white blood cells. Again, 17 known families with multiple members within each family. And interleukins most often work in conjunction or combination with other cytokines and have a whole array of different effector functions. Last is the tumor necrosis factor. This plays a very strong role in the inflammation response or inflammatory response. Its primary activity is to destroy tumor cells. So tumor necrosis factor is usually released. It is produced by cytotoxic T cells and natural killer cells, and they will release the tumor necrosis factor to break down and destroy tumor cells. Tumor necrosis factor can have a regulatory effect on healthy cells and inhibit them from entering into mitosis to try and slow down cell proliferation, which is a common initiating factor in cancer. Another important form of cytokine, and it's really more of a cell receptor than a signaling molecule, but those are the adhesions. Adhesions, there are 4 different classes of adhesions. There are cadherins, which are used in cellular junctions. There are selectins that are found on the surface of white blood cells. White blood cells in venous tissue will stick to the interior of a vein in order to receive other signals where there may be damage or infection. The Ig superfamily supports neural connections. So these are found in nervous tissue. They're called Ig superfamilies because they closely resemble antibodies or immunoglobulins, but they are not. And then last are the integrins or integrins. The integrins are membrane receptors that are found in cell-to-cell interactions, and we are going to see integrins and selectins play a big role in the inflammatory response, and the two selectins and integrins working together are going to initiate changes in cadherins. So adhesions are considered part of the cytokine family simply because 3 out of 4 of them are highly associated with inflammatory responses. So just to review this section real quick, how do the different cells communicate with each other? Make sure you understand the difference between a ligand and a receptor, and also how they respond or how they react together. How are they connected, and what is the effect of them reacting together? Know your cytokines and the 3 classes of interferons, the 4 major families of adhesions and what they do. Are they related to immune function, and how are they connected to each other? We're going to see more about those adhesions, and we're going to be talking about all of the different cytokines throughout the entire chapter, both in the innate immune system as well as adaptive and hypersensitivities. And finally, think about the different cytokines you just learned about. Could we use any of those as some form of immunotherapy? And if we could, how would that work? What would we use it? What type of immunotherapy would we use it for? So I hope you enjoyed our lecture on cell communication. I will see everyone in part 5. Have a good one.